We're on module 5. You heard quite a bit of material, and today we're going to talk about pangenomes and how you can compare a number of genomes using the methods you've learned and using the knowledge you've gained in the last few days. So the learning objective for today is by the end of this lecture, you will appreciate the genome diversity of bacteria. You'll understand the concept of uh, pangenome, and also will examine the patterns that emerge from pangenome analysis and show how they're used to characterize bacterial population structure and dynamics, and also know how to construct pangenome. And in the hands-on the lab tutorial section, then you will get a chance to work with some of the tools. So when we compare any two human beings, they share 99% of their genome sequences and 97% of their genes, if you counting all the duplicates and all the pseudogenes. Or if you're only counting protein coding genes, then that's greater than 99%. Right. So on the other hand, if you take three E. coli strains, what percent of the genes are shared by all these strains? So if you did not look ahead of the slide deck, perhaps I can get a few people to just input on Slack what their guesses are, and we'll take a look. And also while you're at it, maybe you can guess what percent of the E. coli genes are shared by all E. coli strains. So this is, of course, a rough percentage. And what percentage of the Pseudomonas aeruginosa genes are shared by all um, all the strains. So let me see if I can get to the. Okay, so I'll give everyone maybe another 30 seconds to enter your answers. So be a little, so if you're be a bit specific which percentage you're referring to. So um, so let's say three E. coli strains, I think Ryan said 50%, and three E. coli share 99.1%. Is that a guess or 60%? These are all way too high. Anyone else want to guess a lower number? If your own prices is right, I think you're all lose the game. So 5%, 25%. Okay, so the lowest one seemed to be 5%. So let's take a look at what we actually learned from studies. So in 2002, Welch and al. Compared three E. coli strains of different etiology. So one is a uropathogenic strain, one is a enterohemorrhagic strain, and another is it's a laboratory strain that's non-pathogenic. So the K12 so-called reference strain that was sequenced earlier on. And the differences in the gene content is shown on the in this Venn diagram on the right here, right? So you can see that the um, overlapping part of all three strains is only 39.2%. And this is just by comparing these three strains, a bit very different strains of E. coli. Right. And then the differences can be attributed to the different quote unquote lifestyles of these strains. So, for example, the uropathogenic strain is rich in fimbrial adhesions and autotransporters and iron sequester systems, things that are useful in the urogenital tract. Um, of the host, right? So that's back in 20, 2002. And if we fast forward uh, by about uh, 20 years or 15 years or so, when we compare genomes of about 10,000 E. coli, even though they form clades like you can see in this highly compact tree on the right-hand side, uh, only 4% of the genes that are found in any of these E. coli are actually found in all E. coli. So that translates to about the you can overall found about 55,000 different genes in this 10,000 E. coli, and they only share about 2,000 of these genes. Okay, so that's roughly 4%. And you take another extreme case of Pseudomonas aeruginosa, which is a pathogen that's associated with cystic fibrosis patients and also with and it's an organism that's able to pick up and develop resistance to antibiotics, so it's a multi-drug resistant pathogen. Only about 650 genes are found in roughly 1,300 uh, P. aeruginosas studied. So this represents less than 1% of all the genes found in um, Pseudomonas aeruginosa. So the actual references is listed here. So as you can see, most of you have underestimated the diversity of these uh, bacteria. So such huge diversity can be attributed to several evolutionary forces that drive a microbial genome evolution, including mutations, genome reductions, gene duplication, genome rearrangement, and as you have learned already in the previous lecture, uh, horizontal or lateral gene transfer. Okay, and the goal of comparative genomics, therefore, is to identify genetic and genomic variations and study how these variations can affect the functions of an organism. For example, as we have seen in previous modules, these gene variations can make a bacterium more resistant to certain antibiotics or more virulent. And if you are taking the workshop next week, then these variations can also be used to track the transmission of pathogens and, of course, to understand how these uh, organisms evolve. So these genetic variations can be classified into regional, gene, or nucleic acid level variations. So the regional variations is essentially a stretch of DNA that's present in one isolate but absent in others. So these, we often call them as strain-specific chromosomal regions or strain-specific plasmids, or they could also be some sort of tandem repeats. Uh, the gene level variations will be genes that are missing or coding for different amino acids in one isolate compared to another. So these are so-called strain-specific genes. They're different alleles of a gene, and they can also but we are only looking at genes and not the regulatory elements and the non-coding genes. Nucleic acid level variations can be as small as a single nucleotide difference in one isolate compared to another, and these are commonly referred to as single nucleotide variants or single nucleotide polymorphism. So we looked at an example of a three-way comparison of E. coli published in 2022. However, the honor of the very first bacterial comparative genomic paper goes to Elm et al., published in 1999 of two Helicobacter pylori genomes. Um, so what they found is that more than half of the strain-specific genes are clustered in hypervariable regions, shown as red, point out as red arrows for these blue regions here. And and these regions are commonly, as you've learned, referred to as islands or genomic islands. And this type of observation soon was regularly observed in many other species. So if you recall in modules we talked about, if you have a stretch of uh, DNAs that have different characteristics or that looks different compared to the rest of the genome, it could uh, be due to horizontal gene transfer. So if we fast forward by 25 years to today, and now we have millions of microbial and viral genomes available in public repositories, and the comparison of these genomes is now routinely done in order to understand microbial evolutions and functions. So I'm highlighted a few databases that might be familiar to you, such as the NCBI microbial genomes database. Uh, there's some um databases that have contained assembled and processed genomes of a huge number of bacteria from this group of researchers here. And also there are viral databases such as during the COVID and to date, roughly 17 million SARS-CoV-2 genomes were sequenced and deposited in public repositories that you can either have direct access to or have to apply access to. Um, and indeed, we gain new knowledge and new understandings of these organisms through the comparative analysis, and having access to all these microbial genomes is very helpful to the community. So if you think about comparative literatures or comparative art, indeed, often we learn new things and get new ideas and new insights through comparison. So here's a bit of a public announcement that hiding, which is not directly related to today's discussion, but hiding behind the success of these huge number of genomes is the fact that the data are not very reusable and don't contribute to precision public health. In other words, ability to look at large number of genomes across very different settings or countries collected by different people. And here's an example of this cassette database of viral genomes that I mentioned in the last slide. So you can see from this study that they reported 63% of the sequences missing demographic information, and 84% don't have sampling strategies, meaning that we can't use the data properly for epidemiological modeling. If you don't know the sampling condition with the study strategy, then it's hard to do epidemiological interpretation. Um, and the and 95% of these sequences don't have patient-level clinical information, so we can really use the data to associate sequence variations to clinical outcomes and therefore use it for, um, for example, for treatment development or vaccine development. So similar level missing contextual data is also found in microbial genomic databases. Right? So this is something that we'll talk more about next week in the other workshop, in case you're taking it. So when we do comparisons, we usually treat one example in the group as the reference and then compare all the other examples or the other samples in that one group to the reference. Um, However, when you know that there's such high diversity observed in bacterial genomes, the sequence of a single genome is not sufficient to reflect the genetic variabilities within the even within a single bacterial species, right? So picking a reference genome to represent the species then is not possible or not ideal, right? So comparing strains of interest to a single reference can create analysis that are biased by the reference you select. So it's shown in this diagram here, right? If you take a traditional reference-based approach and your query sequences have some additional DNAs, then it's quite possible that those additional DNAs cannot be aligned to the reference and therefore potentially are lost in the uh, analysis process, right? So we need a reference-free approach or representation to um, reduce the bias. Okay, so this is uh, when the concept of pangenome come into play. So the pangenome idea was born out of comparative genomics that revealed high variations among strains, and also the researcher there at that time was interested in reverse vaccinology studies. In other words, using genomic data to identify vaccine candidates. So the term was first coined in 2005, at least for microbial pangenome, in a paper by Hervé Tattelin and al. From the Institute for uh, Genomic Research. This is led by Claire Fraser at the time, who actually was my postdoctoral supervisor. So while I wasn't there during that time as part of when this work was underway, in subsequent, so I got there in about 2007, I think. Yeah, and so I went there, I got a chance to met Hervé and Claire and work with others on metagenomics or microbiome studies, I should say. And through osmosis, of course, we heard quite a bit about pangenome analysis and development at the time. And you can see in the picture here that this is um, uh, pictures of Hervé here and here and some other people's in the group. And there's a recount of the pangenome development process in this article here that I linked and draw a lot of the material from. So it's a great read if you want to learn more about the development of microbial pangenome. So essentially, pangenome is defined as the entire repertoire of genes accessible to a species, right? So no longer looking at a single reference, but looking at a collection of organisms from a single species to form the collective pangenome. So the pangenome is typically divided into three main components: the core genome, which are genes that are shared by all strains of the species. Uh, these usually include essential housekeeping genes for basic cellular functions. Then they represent the evolutionary backbone of of a given species. And then you have the accessory genome, which are genes that are present in two or more strains, but not in all strains, and they can be acquired through horizontal gene transfer in the past and propagated down a lineage, right? Um, so they often associate with adaptation to specific environment and things like antibiotic resistance, virulence factors, or increasing the metabolic flexibility of the species as a whole and of that particular lineage that have these accessory genomes. Uh, then the last class is the unique genes. These are strain-specific genes, and they're typically acquired through horizontal gene transfer or more recent mutations, and often linked to niche adaptations or, in some cases, to pathogenicity. They also consist of many hypothetical genes of no known functions. So the question posed initially by Tadlin et al. Was how many genomes from isolates of the group B strep species do we need to sequence to be confident that we identify all of the genes that can be harbored by this species as a whole, right? So instead of comparing all strains to a single reference, as you see in this diagram here, pangenome analysis does an all against all comparison, so seen on the right side here. And this is what referred to as the reference free. You don't no longer rely on a single reference. And what the researcher observed using these 8 group B straps was that as they extrapolate the curve that's formed by the average, and later on they use the median number of new genes found within each additional genomes to the existing N genomes that's already considered, the curve never reached zero. So this is shown on this graph here. So each of these dots essentially represent a comparison. So what we of the 8 genomes, essentially you have 2 that's already you consider that as a quote unquote reference you're comparing to, and each of the 8 then is in turn compared add uh, to the 2, and then and you do that for all 8, right? And then the next iteration you see here is that you start with 3 genomes and you then treat the other 5 as new genomes. And essentially you plot the number of new genes identified as each uh, genome is added to the pile. So when they went through that exercise, they basically observed that this curve extrapolate but never reaches zero. So this means that there are always new genes expected to each new genome sequences added, no matter how many genomes you already have. So this led to the idea that a pangenome can be open or closed. So pangenome openness is a concept that describes how the size of the species pangenome change as more genomes are sequenced. It reflects the species' genetic diversity and its capacity to acquire new genes, right? And we know that different species or different organisms have different capacity in their ability to acquire new DNA through one of the mechanisms of horizontal gene transfers. So an open pangenome keeps expanding as more genomes from the species are sequenced. So it indicates high genetic diversity and frequent horizontal gene transfer. In highly open species, the core genome can shrink dramatically over time. Right? So the red arrows here shown as a depiction of open pangenomes. You can see gene count of core continue to grow, and the number of core genes, as we've seen in E. coli and Pseudomonas examples, represent a very small percentage of the overall gene pool. On the other hand, you have closed pangenome that reach plateau after sequencing a certain number of genomes, meaning a few or no new genes are discovered after a certain number of genomes are sequenced. So the core genome size stabilized quickly, um, as you can see depicted in these blue arrows or curves. And this is because most of these strains share the same gene set, and the core remains relatively large and compared to the open species, open pangenome species. Now, the rate of new gene discovery per new genome can be mathematically described using what's called Hip's law. So it's a power law. And essentially, the details is actually discussed in the reference I mentioned earlier, and also again in here, for those of you interested. But for the sake of time, I won't get into the detail of how they actually found a formula that best fit the extrapolated curve. Okay, so in other words, if the number of strains needed to capture the pangenome of a species is finite, then we consider that species to have a closed pangenome. Otherwise, it has an open pangenome, right? So it's shown in the figure on the right here. The same group went on to do a study, compare other species genomes, um, and what they discover is that uh, as a function of the number of new genes discovered, um, some organisms, unlike the group B strap shown in the middle here, indeed have a trend line that reaches zero eventually. So in the case of B and Bacillus anthracis, um, the causative agent of anthrax, you can see that it's will be considered a closed pangenome, and after about 7 or so genomes analyzed, they didn't find any additional new genes. So this type of concept then help microbiologists to predict how a species will evolve over time. Is it likely to acquire many resistant genes, or is it a genome that's under reductionist uh, trend, and it's actually becoming more niche adopted and so on. Okay, so however, due to horizontal gene transfer, there is no absolutely closed pangenome. So over the last decade or so, as more genomes for each species were sequenced, indeed, most of these closed pangenomes that were reported by earlier efforts with smaller sample size and also uh, often not considered environmental isolates, we were uh, able to revise these closed pangenomes to be open pangenomes. So some of these examples include Staph aureus, which was discovered to have extensive repertoires of mobile genetic elements. So it was revised from closed to an open pangenome. Listeria monocytogeny, which you have come across in earlier modules, um, also was revised as environmental sampling of this pathogen identified new diverse set of genes that are part of this species repertoire. And B. anthracis and also Mycobacterium tuberculosis, the causative agent of tuberculosis, on the other hand, still have a relatively closed pangenome. So they're relatively recent clades, and they have limited MGEs. Um, so they get cautionary, cautionarily renamed or slowly open pangenomes. So in short, species that have isolated niche adaptation that have undergone genome reductions or are newly emerged lineages are more likely to have these sort of closed or slowly open pink genomes. Okay. Okay, maybe I'll pause briefly here just to see if there's any questions up to this point. And I can see.
We have a
Okay,
hands
I
up
can see
from
the Slack.
Shamsuddin.
So that's fine. But if there's any raised hands, let me know, Neha. So, do you think it's possible that two different species that live in the same environment will have higher genetic similarity compared to similarity between the same species that live in very different lifestyles or environments? Um, that's a good question. I don't have a specific example that come to mind, but certainly I would expect that to be the case that um, organisms that found in the same niche, well, let's just say within a species that found in the same niches, or I guess in this case, you say two different species, right? So um, it depends a little bit on their relationships, right? So often if they are competing within the same niche, then they could have shared certain genes that are needed to metabolize the nutrient that are found in that environment. But they often can also be cooperative. In other words, they assume different roles in that niche, right? So, and that's very common even for eukaryotic systems, right? You have prey, you have predators, you have animals assuming different niches, different lifestyles, and so on. So, from the same environment, uh, if it's two different species, then they, depending on their, again, their interactions, they may not actually have uh, more similar genetic uh, composition or set of genes compared to same species, very different lifestyles. Hopefully that makes sense.
Will we have another question from Shamsuddin? Go ahead.
Okay, go ahead.
Thank you for the brilliant lecture. Um, I do give lecture to medical students on leprosy, and I know that the genome of leprosy has lost so many genes over since it diverged from tuberculosis. And
Mhm.
I know, yes, and I know there isn't much horizontal gene transfer, at least that I know of, with leprosy, with lepre. So I'm assuming lepre would probably be a closed genome or maybe slowly open genome. Thank you.
I believe so. Yeah, that's my recollection as well. I think so. So yeah, we can verify that by afterwards, but I think based on my recollection, that seemed to be a case, and that also applied to some other highly niche adapted genomes that have lost its that's just highly specialized in an environment and doesn't seem to be picking up new genes.
Gary has his hand raised.
Yeah.
Okay, great.
So I just wanted to just weigh in, and I'm agreeing with Will and confirming that for Mycobacterium leprae, it's was probably at one time almost certainly a free living organism, or at least like a facultative, which means it can live in the open, or can invade a host like us, and that's what it did. And over time, it's been adapting to living in that host environment and increasingly becoming like an obligate um, parasite lifestyle. It's shedding the genes that it doesn't need anymore, but it does it slowly. So it is actually does not. It is kind of like gets. So what happens is it's getting sequestered away inside of these cells, and it's not really interacting with other um, bacteria in the community, in the colony that can share their genes. And so you're right, it's not really engaging in any horizontal gene transfer. It's also because it's in a stable environment, there's not really much evolutionary pressure. So the genome is evolving slowly, very slowly. And so that's why we kind of see it in this process of deleting its genes, making pseudogenes. And then over time, it will shed those genes. But it almost certain with I haven't checked, but you know, I would bet like a year's salary that if you did the study, you would find that it has a closed genome.
Yeah, thanks, Gary.
Okay.
You can keep your salary, I'm pretty sure.
Okay.
Uh, okay, so now let's move on a little bit. So we also, when we look at pangenomes, we also need to consider the effect of non-random sampling and sampling bias, right? So there's a quote from the article I cited earlier that as in any population modeling exercise, the conclusions at the population scale are heavily dependent on the randomness of the sample, particularly if it's a small sampling size, right? And uh, can be seriously affected by the presence of structure in the population. So, for example, in our current microbial genomic database, the database is highly biased towards medically important organisms and clinical isolates. And also, geographically speaking, more samples are from the global north countries rather than the global south, right? So these biases can interfere with how we construct and interpret pangenomes. So if a few related isolates were sampled in an otherwise heterogeneous population, this will underestimate the population diversity, right? When construct a pangenome, some studies, on the other hand, err on the side of maximizing the diversity. For example, only sample one genome per clade or per type, like for example, per serotype, then this may actually overestimate the pangenome diversity, or rather, it really just maximize the genome diversity and give a false impression that this uh, organism is more diverse. So when constructing a pangenome, um, uh, these type of sampling biases need to be sort of kept in your mind as you interpret the data. So and also this then also affect how we interpret the result also affect how we interpret bacterial species. So there's another quote that says the very idea of an unbound genome repertoire for bacterial species was opening the microbiology communities to a new way of looking at bacterial species and their anatomy, right? So species that were thought to be a definitive concept with a very defined set of genes no longer is true in micro organisms. So species that were have been molecularly defined as sharing 70% of the genomic DNA based on DNA to DNA hybridization, or at the sequence level relatedness. These type of thresholds no longer apply, as you've seen in earlier lecture, right? So, but we still retain the concept and the term species because it helps to ease the labeling of a group of organisms, kind of to give them a name so you can uh, talk to, we as humans can talk to each other about a group of organisms, right? But the fact that functionally the presence of a core genome that is shared by the group of organism also help to justify that species concept as a working model, right? So we should appreciate that species boundary is fuzzy and that some species are very diverse. They're open pangenome and some are more stable and have closed pangenome, right? And because microbial species is a man-made concept, the pangenome concept that initially developed in the species context can actually be applied to an arbitrary clade of organisms at different taxonomic rank. And we'll talk about clades and phylogeny and so on in the next session, but can just think of a lineage or a group of related organisms can be used to uh, do a pangenome analysis. So it's also worth appreciating that pangenomes now is a common representation for eukaryotic genome as well, including the human genome, as I gave a paper link here. So the ability to directly look at the variations rather than just relying on a reference genome, and the ability to appreciate such divergence and adaptation is a powerful concept, right? So it has been uh, applied to all sorts of organisms beyond microbiology. So now let's switch gear a little bit and look at how we can construct these pangenomes and what the general approach is. So originally, the gene-oriented comparison was used, right? So it ignored the intergenic regions, and this can be done using a pangenome matrix, as we see in the next slide. However, later on, most tools that have been developed use graph representation as the native format. So these are usually termed pangenome or genome graph. So genome graph allow base pair level representation of the variations rather than just gene by gene variation. And also some tools in order to speed up and to reduce the computational resources needed use exact k-mer matching, so alignment-free based approach to build the graph representation. Okay. So I think you cover k-mer and some of these um, the concept of alignment and concept of exact matching. So I won't uh, go over those here. So again, courtesy of Gary's sharing some of his slides with me, this is an outline of how you will construct a pangenome, right? So first you will collect a set of genomes, again, in hopefully a less biased manner. And hopefully these genomes are high quality and you can actually get good assemblies out of these reads. And the more diverse the strains, the better it represents the species genetic diversity, right? But as I mentioned, it's a balance between essentially that ideal of randomness picking, and so you more versus more purposeful picking of trying to pick, say, one representative per lineage. Right? Okay, so then the next step is to then predict the genes and annotate them using these annotation tools that you've seen earlier and standardize. It's important to realize that standardized annotation is important because now you're comparing across all these genomes. So you want to annotate them as consistently as possible. So the same gene is called by the same name rather than by different names if you annotate with different tools. So it's often important that when you do pangenome analysis, you don't just take the existing annotations from public repositories. So you actually go through the process yourself and be more consistent about the annotation. So in other words, if you introduce bias in the annotation, at least it's consistent across all the genomes you're analyzing. Right? So then you want to identify the homologous genes. So compare all the predicted genes across the genomes and group them into gene families. And this can be done using sequence similarity tools and clustering algorithms. And as you can see here, the gene family is represented on the y-axis. So, and then the genomes is on the x-axis. So essentially, this is saying that the gene X is found in all these genomes, so it's considered the core, right? And ones that are partially found is accessory, and ones that are only found in one genome would be considered unique. Okay, and now you have the pangenome matrix, then you can create a visualization that show you the presence or absence of these genes across all the genomes. And this can be achieved using one example is this tool called MVO, but you can which gives the nice sort of track-based display, and this allows you to visualize core versus accessory genome, and you can also construct phylogenetic trees and look at the gene content and so on and so forth. Okay. And there are different tools to achieve this same task. So found a paper that summarized or compares 16 different tools, all doing these similar approach of gene oriented comparison. And then you can see that they have different they use different method and different functionality. So it's a good reference paper if you are interested in understanding the pro and cons of the different tools. So the second approach is a graph-based approach that all the genomes are combined into a graph structure where the nodes are essentially the sequences. It could be a k-mer, it could be a gene, or it could be a unitig, right? A DNA fragment that's unique in the sample. And the edges, on the other hand, are just connecting these sequences. But some representations can actually use the edge to encode the variations. So you have to be careful when the different, even though it's all graph-based approach, the graph representation, the graph structure can actually be different depending on the algorithm. Okay, so the first type of graph representation I want to talk about is this color de Bruijn graph, which we will use in the tutorial. And in this case, the genomes are decomposed into k-mers, as you can see here. And the adjacent overlapping k-mers then are represented as a graph showing showing it here on the right hand side here, and the strain information then is encoded as colored associated with colors are associated with these nodes. That's why it's called color de Bruijn graph. And this is to show which strain have which sets of k-mers. An example of a tool like this is called Bifrost, and which you'll use in the lab today. And as you can see here, the different path in the graph represent the different strains or different sequences here, right? So, and you can also see that as you add new strains, the information can be overlaid on the same graph. So it's fairly memory efficient, highly scalable, right? The second approach can be done either by compacting or combining the adjacent k-mers that are invariant across strains, right? So if you see a stretch of sequences that are which are longer than the k-mer but are invariant across strains. I think there's some examples in the first part here, right? For at least for those two. Then and these invariant regions essentially represent the core or the identical genes that are across all the strains. then the alternative path then show the variable sequences across the different strains, right? So it's as shown in the second graph here. The first part is identical, and then the second part has variations in different strains. So this can be achieved also by alignment of genes and DNA fragments rather than by merging of the fixed k-mers. But this then require alignment, which is a lot more computationally intensive than exact k-mer matching based approach. And therefore, this approach is less scalable. However, it does it can provide a more compact representation, as you can see here. And also, each of these know then it's more biologically relevant representation. Okay, so an example of this type of graph is pen graph. Okay. So here's a recap of some of the common tools for microbial pangenomes. So Rory is one of the first scalable gene-oriented pangenome construction tool, one of the first tools, I should say. And it's still in use, quite popular today, but it's not really maintained. So there's a sort of improved version called Panaroo using a graph-based approach to clean misalignments or misannotations and frame shift and other errors. That's also does the gene-oriented comparison. On the other hand, I mentioned Bifrost already. It gives you the scalable k-mer graph suited for huge set of genomes. And however, the post-processing, as you see in the lab, is needed if you want to match a gene feature to the k-mers. And then the pen graph is, on the other hand, gives you a biological, more meaningful pangenome graph, but not ultra scalable. There's a whole list of probably hundreds of different tools available from this link covering both eukaryotic and prokaryotic pangenomes. So you also should know that some of these algorithms are, and most of these algorithms are actually greedy, and therefore the order of processing the genome can result in slightly different set of genes, gene clusters, right, gene families, or also different members within a cluster. So just be careful that, if you if you sort the genomes randomly and run it through the pipeline of these some of these tools, you can actually get slightly different clustering or slightly different gene family memberships as a result. Okay. So lastly, I want to quickly mention that there's another visualization tool called Bandage that you'll use in the lab. It's initially developed for visualizing de novo genome assembly. However, and it allows you to zoom in and out of the visualization, and you can also do blast search to match the genes to the k-mers, as I mentioned earlier. Okay, so now moving on to final part of this, so probably another 5 or 10 minutes and we'll be done. I know we're approaching the end of the hour. So I'll end the lecture by giving an example of pangenome analysis that Jimmy from my group did in collaboration with the Prairie Diagnostic Services in Saskatchewan. And in this study, what happened was that they noticed a sudden increase of death in grower pigs from the same nursery farm. And the post-mortem examination then revealed that there's inflammatory lesions in multiple body parts. And they isolated several different types of bacteria from these pigs, as you can imagine, they're not just going to be a single clear pathogen. Often there are multiple um, or pathogens or potential pathogens, let's call them, associated with these samples, right? So so they then send 6 of the isolates of Salmonella to the reference lab in Guelph for Illumina sequencing and in silico serotyping. So all of them actually returned Salmonella derby of the same sequence type 40. And as you can see on the graph on the right here, sequence type 40 is the most common sequence type in the database, right? So many people might just give up then and say, okay, this is a common um, sequence type, so there's nothing really special to make it hypervirulent. But Jimmy actually persisted, and the people at PDS. So first, he compared the isolate sequences against PulseNet Canada databases, and then placed the cases in the Canadian context, right, to try to understand where these isolates might be coming from. Where are they different from the known from other isolates that have been studied before or been isolated before? Okay, so this is a tree. Again, you'll learn about tree constructions and so on in the next session. But indeed, what they found is that the hypervirulent S. derby isolates were clustering together and form a distinct clade by themselves, as shown with over here, and also zoom in. You can see that these cases are all most similar to each other and form a clade here. Okay, so they're interested in characterizing the isolates further. So the first thing then is to see if there are um, isolates or strains that can be used in comparison going beyond the 6 isolate and as a comparison, right? So so we use a tool developed by Jimmy again from our group called SAMTrack that essentially was able to search against the entire NCBI database of Salmonella genomes, over 400,000 genomes, very quickly, and then identified closely related genomes. So 107, I believe, were identified, these positive matches. These matches together with their case isolates are then used to build the pangenome graph using Bifrost. And genetic variations in the homologous regions then result in alternative paths in the graph structure, right? In other words, they can be aligned, but they're not identical, so they form a bubble in the graph, right? So then, as a result, they were able to I'll just switch to the next one, sort of hone in to using the color de Bruijn graph, hone into specific variations that may be of interest, right? So by comparing the case isolates with the closely related strains, right? The reason we didn't compare across a wide range of salmonellas is that we want to hone in on the differences that explain this particular 6 cases, why they're hypervirulent, rather than if you include a diverse range of isolates, it's going to add noise to your comparison, because unrelated unique sequences is also then going to show up, right, in doing the comparison. So they zoom into this region here. It's in the coding gene part of the RRC family of transcriptional regulators. And what Jimmy found was that there's actually a non-synonymous point mutation in these case genomes, and it resulted in a premature stop codon that truncated the gene, right? So this is what a pseudogene is, and indeed the annotation of the case genome, the sepsimia cases genomes, label this particular open reading frame as a pseudogene compared to the reference, which has an intact copy. Okay. So then a bit of literature search showed that this family of regulators are involved in complex feed-forward and feedback loops to control the expression of a type 3 secretion system in Salmonella pathogenesis, specifically in SPI or Salmonella pathogenesis island 1. And this type 3 secretion is involved in pathogenesis. Okay, so it was then conjectured that the pseudogene actually led to a deregulation of the regulatory pathway here, and as a result of that deregulation, the expression of the type 3 secretion apparatus or the genes are then increased or upregulated, resulting in the hypervirulence. So this is something that we're actually currently testing out in collaboration in a lab. So I just want to end by saying that there are still challenges associated with pangenome analysis. So sampling bias and population structure can lead to misinterpretation of the pangenome, and high diversity of microbial genomes can lead to bioinformatics data processing and data analysis errors. So this includes assembly errors, alignment errors, annotation errors, and other errors such as ortholog clustering errors and so on. So it's also worth keeping in mind that most of the genomes that we're comparing to are incomplete, right? So you're never fully sure that the unique genes are truly unique, or even the core genomes that you identify is actually core if or just happen to be missing in one of the strains. So and also these genomes, if they're not high quality draft genomes, could contain sequencing errors, or rather, if they're not complete genomes and they're draft genomes, they could contain sequencing errors. So recall that pseudogene example I just mentioned. This would have likely to be missed if you have a low quality draft genome where the mutations didn't stand out so saliently, right? So horizontal gene transfers, we learned, further complicated the phylogenetic signals and add to the analysis complexity. So this is actually well summarized in this particular article here. Okay, and that's it for my lecture.